Hello everyone. Today's topic is lymphoid organs. First we know about types of lymphoid organs. There are two types of lymphoid organs. Primary lymphoid organs provide appropriate microenvironments for the development and maturation of lymphocytes. In other words, these are the places where immature lymphocytes become committed to a particular antigenic specificity. Thus, immunocompetency is attained in primary lymphoid organs. Example of primary lymphoid organs are thymus and bone marrow. Secondary lymphoid organs trap antigen from particular tissues or vesicular spaces. In other words, it can be said that secondary lymphoid organs provide site for antigen-antibody interaction. Here is the pictorial representation of human lymphoid systems in which the primary and secondary lymphoid organs are highlighted. Now come at thymus. As shown in the diagram, thymus is located just above the heart. It has two lobes, that is, it is a bilobed organ. Each lobe is divided into many lobules, which are separated from each other by trabeculae. Trabeculae are strands of connective tissues. In this diagram, only a single lobule is considered. It is divided into two zones, an outer cortex and an inner medullary zone. In the outer cortex area, densely packed immature T cells are present. Nerve cells are also present in the cortex regions and they surround the thrombocytes, forming a large multicellular complexes. There are very few thrombocytes in the medullary region. Epithelial cells, dendritic cells and macrophages are present in both the cortical and medullary regions. Function of thymus is to generate and select a reservoir of T cells. Maturation of thrombocytes generate an enormous diversity of T cell receptors. T cells having receptors that can recognize antigen major histocompatibility complex are retained by the thymus while rest of the T cells die by the process of apoptosis. Those T cells that have self-reactive antibody receptors are also destroyed within the thymus. Regression of thymus occurs with age. Thymus attains its maximum size at puberty. As is visible in the diagram, its size increases after birth, but at 10 or 12 years of age, the size gradually decreases and after the age of 60, the weight of thymus is reduced to around 5 grams. Bone marrow. It is a spongy tissue present in long bones. As is visible in the diagram, the yellowish part is bone marrow. In this diagram, hematopoietic cells are present. They are responsible for B cell proliferation and differentiation within the bone marrow. Stromal cells present within the bone marrow interact directly with the B cells and secrete some cytokines responsible for B cell development. Function of bone marrow is origin and development of B cells. There are few things to be noted in case of bone marrow. B cell proliferation occurs in bone marrow only in case of mice and humans. In case of birds, bursa of fabricus is a site for B cell maturation, while in case of cattle, ileal Peyer's patch is responsible for B cell proliferation. In rabbits, appendix take up this work of B cell proliferation. In the next video, we will talk about secondary lymphoid organs. Till then, thank you.